So we are able to hear because we have the organ of cortex. The trick is to move the hair cells right here. In order to do that, we need to move the basilar membrane. When we move the basilar membrane, the hair cells are going to move because the tectorial membrane is basically fixed. For example, we produce sound waves with our vocal cords. The sound waves go through the air and they go inside our auditory canal. They bounce against the tympanic membrane. The tympanic membrane vibrates. That vibration is transmitted to the malleus, incus, and stapes, and the stapes is connected to the oval window that is going to transfer that vibration inside your cochlea right here. Now, in the cochlea, the vibration of the three little bones is transferred inside into the fluid in the form of waves. The waves are going to go through the perilymph right here, and that perilymph is going to move the vestibular membrane. The vestibular membrane moves the endolymph in the cochlear duct. The endolymph moves the basilar membrane. By moving the basilar membrane, we are going to move the hairs. The hairs open channels for potassium, and that initiates a depolarization that sends the information to your brain using this nerve, which is going to be the vestibular cochlear nerve. The basilar membrane is stimulated in different areas to allow us to hear different sounds. The vestibular cochlear nerve to send information to the brainstem, from the brainstem to the thalamus, and from the thalamus into the primary cortex auditory area. Our next topic is equilibrium. There are two main types of equilibrium: static equilibrium and dynamic equilibrium. The macula is for static equilibrium. Semicircular canals are for dynamic equilibrium. In the vestibule, we're going to have structures called macula. If you look at this figure right here, what we need to do is we need to move the hair cells. For example, when you're inside your car, you don't move, but the car moves. The car movement will move your macula, utricle or saccule, and then this gel right here will lag behind, and because it lags behind, it will move the hairs. And when it moves the hairs, transfer information to your brain. The semicircular canals. In this case, semicircular canals is when you are turning. For example, when you are sitting down on a chair and that chair starts turning around, then that's when you use the semicircular canals. The semicircular canals have a gel. In this case, it's called crista ampullaris. And inside the gel, you have hair cells. What happens is that when you turn, the fluid inside this tube is going to push the crista ampullaris, and then this pushing moves the hair cells, and when the hairs move, they send the information to your brainstem, from there to the thalamus, and from there into the vestibular cortex that is going to help us maintain equilibrium. Don't forget that we have three different areas to maintain equilibrium. One of them is the eyes, the other one is the cerebellum, and the last one is going to be the ears. Vision, we're able to see light. Vision is the perception of objects in the environment by means of the light that they are going to emit or reflect. If there is no light, we cannot see. So right here you have the eyes. The white part is the sclera. The color of your eyes is going to be the iris, and pupil, the black part in the middle. The iris and the pupil are covered by this crystal and translucent area called the cornea. Also, right, we have the lacrimal glands. The lacrimal glands produce a lot of tears to clean your eyes. It also has substances such as immunoglobulin A in order to defend us. The tears come out of the eyes through the lacrimal sac into the nose through the nasolacrimal duct. In addition to those structures, we are going to have muscles associated with the eyes. So this is superior rectus down here, inferior rectus, medial rectus, lateral rectus. On the top is superior oblique, and another oblique at the bottom, inferior oblique. This muscle moves due to the fact that they are associated with nerves. In the case of superior oblique, it's the trochlear nerve. Lateral rectus is the optic nerve, and all the others are going to be moved with the oculomotor nerve. We can see the anatomy of the eyes after the eyes have been cut, so you can see the inside. So there is a space called the anterior chamber between your cornea and the lens, where you're going to have the aqueous humor. Behind the lens, you have this other space where you're going to have the vitreous humor. This is the wall of your eye. Okay, it has three layers: external, middle, and inner layer. They are called tunics as well. This external area is going to be the sclera. The middle area, so called the uvea, is going to have a lot of blood vessels, and it has three main parts: the choroid, which is this part in brown, which continues with the ciliary body, which is going to be the second part, And the third part is going to be the iris. The ciliary body produces this muscular mass that is going to be able to hold the lens in place using the suspensory ligaments. So what's the job of the lens? The lens sits behind the pupil and it allows to adjust the light before it reaches the back of your eyes. So the light goes in through your cornea, through your lens, vitreous humor, and goes to the back of your eyes. The inner layer, right here in yellow, is what we're going to call the retina. The retina is where we have the receptors that will allow us to see. In your retina, this is the area where the nerve is, and this area right here is the area that will allow us to see detail. So where the nerve is located is called the optic disc. This area is orange, and this will allow us to see everything. This is what we consider the peripheral vision. This area where all these receptors are going to concentrate, they're going to be the macula lutea. The very dark area right here is going to be the fovea centralis that is going to give us the detailed vision, right? Or the ability, for example, to read. So this is peripheral vision. This is visual acuity. So these are the receptors that you have in your retina. The rods and the cone. Cones is for a lot of light. Rods are for a little bit of light. Cones are, for example, during a sunny day, and rods are, for example, for spaces where there's not a lot of light. If there is no light, we cannot see. In addition to that, there are other cells such as ganglion cells and bipolar cells. Some people can read, for example, but they cannot identify colors. There is a test called Ishihara's test for that purpose.